Hello students, welcome to my class. Today we will discuss the double antibody sandwich ELISA. Double antibody sandwich ELISA. That is the second type of ELISA. Last day we discussed about indirect ELISA, that is the triple antibody sandwich ELISA. Today we will discuss a double antibody sandwich ELISA. This can also be said a direct ELISA because this method is used to detect directly any type of antigen present in the body, or hormones, or drug, or steroid, any type of biomolecules, or any type of chemical molecules, biochemical molecules, or any antigen present in the body or anywhere. In the case of plant as well, it is also used in horticulture and agriculture to detect plant virus. That means it is also used to detect viruses, plant as well as animal viruses, hormones, and steroids, very much used in detection of hormones and steroids, and as well also for detection of drugs. Okay, so this is the utility and use of double antigen sandwich ELISA. Okay, to detect hormones or drugs or steroids as well as antigen, particularly in the plant case of plant. Okay, so this is the use of double antibody sandwich ELISA. Why it is called double antibody sandwich ELISA, we will explain, you will understand. I will explain you through this diagram. Okay, this is the four type of diagram. This indicates, represents the four steps in the process of double antibody sandwich ELISA. In the first case, suppose this is the surface of the well, microtiter well, okay, of ELISA plate, which is coated with an antibody, and the exposed surface we discussed in the last class. The exposed surface is covered with some neutral compound biomolecules like bovine albumin or casein या bovine albumin. Because these molecules bind to the exposed surface and cover it, preventing binding of any unrequired or non-required molecule antigens present in the sample to this, otherwise this will hamper the process. Okay, so in the first case, this is the well, this is the well, this is the antibodies specific to the antigen is bound to the surface, and these are the point where the antibodies are bound in the well, microtiter well, and this is the exposed surface where the antibodies are not bound. This exposed surface is covered with a neutral or inactive, non-reactive compounds like casein or bovine albumin. Binding of these non-reactive molecules prevents the binding of any undesired antigen to this surface to interfere with the result of the experiment or process. Okay, so this is the first step. This is the first step. First step is the preparation of the proper surface of microtiter, that is, it should be coated with the antibodies, and the exposed surface should be covered using bovine albumin or casein like non-reactive molecules. In the next step, the sample containing the antigen. This may be a steroid molecule, this may be a hormone molecule, either protein molecule or a steroid molecule or any type of drugs or any virus in the case of, particularly in the case of plant. Okay? So this is added to the microtiter well. Suppose this is the antigen and this is added to the well. This antigen will automatically, because obviously this will bind to the antibodies attached to the well. Okay? This will, once this is bind, that means a step is that a sample is added and it is incubated for a while, desired time at a particular temperature, required temperature for some time. Okay? This time is given to the antigen to bind properly to the antibodies. After that, the plate is removed from the incubator, taken out from the incubator and is washed. Washing is done to remove any unbound antigens or any other type of things present in the sample should be washed off, to be removed from the well. And what is remains? Only the antigen and the antibodies which is bound to the surface. So only this component will remain in the well. Okay? Which is required. Only these two is required. So only these two required things will remain in the well, coated well, microtiter well, and all the other unrequired, undesired substances present in the sample will be washed off or removed from the well for the accuracy of the experiment. This is required for the accuracy of the experiment. After washing, the next step starts. This which is the third step. Okay? Okay. In the third step, the enzyme coated a specific antibodies. Antibody is specific to these antigen which are here. The antibodies which is specific to these enzymes, these antigens or hormones or anything, these antigens is conjugated with enzyme. Okay? So these specific antibodies which are conjugated with enzymes, this enzyme may be horseradish peroxidase or alkaline phosphate. In most cases, horseradish peroxidase is used. So these enzyme conjugated antibody are added into the well. After the addition of the antibodies, conjugated antibody, the whole plate is again incubated for some specific period of time. By that time, the antibodies is conjugated to the enzyme get attached to the antigen, and only those antibodies which are added to the antigen are required. Rest of the antibodies which is present in the solution is removed by washing. Washing removes the excess antibodies from the well, and only bound antibodies remains in the well, which is bound to the antigen. This is the third step. In the fourth step, the substrate is added. Which substrate? Substrate for these enzymes is added. We have already discussed the type of substrate using the ELISA, particularly for horseradish peroxidase. Okay, so this is added, substrate is added. Once the substrate is added, this will react with the this will react with the enzyme, and enzyme will convert it into a product, and that product is a colored product. So development of color will indicate the presence of antigen, because if the antigen is present, only then the antibody will bind to this and will remain in the well. Otherwise, if this antigen is not present in the well, this antibody will not bind to this antigen and will not remain in the coated well after the washing, because the washing will remove any floating or any unbound antibodies present in the well, and only antibody will remain in the well after washing which are bound to the surface or to the surface indirectly through the antigen. Okay, this is the process, because this is present bound to the well because this antibody is present. On this antibody is responsible for the binding of this antigen and remaining of the keeping it in the well. Okay, and if this antigen is present bound to this antibody, this antibody which is conjugated with enzyme will bind to it and will remain in the well even after washing, because it is stick to the antigen and will not washed off, will not be washed away during washing. Okay, so if color is developed, that means these antibodies is attached to the antigen which is present in the coated well. That gives the idea of presence of antigen in the well. That means this antigen will come from the sample. That sample contains this antigen. That is why the color is developed. Okay, the development of color indicates the presence of desired antigen, the antigen we are looking for. Okay, if the color is developed, that means the desired antigen is present in the sample. If color is not developed, that means the antigen is not present, because at the time of washing all the antibody conjugated to enzyme will wash off, and no enzyme will remain in the well, and even after addition of substrate, no color will be developed, because the enzyme required for their conversion of substrate into product is not present in the well. So that means color will develop only when the enzyme is present, and enzyme will be present in the well only when the antibody is bound to this antigen. Okay, so that means a color develops only when the antigen is present in the sample. Okay, so this indicates the presence of antigen in the sample. This is the qualitative analysis, and for quantitative analysis, the intensity of the color produced in the product is estimated using an electrophotometer, and with the help of preparation of a standard graph, we have discussed preparation of a standard graph, we can estimate the amount of antigen, drugs or hormones or any virus present in the sample can be calculated or estimated. Okay, so this is the process of double antibody sandwich ELISA. Because it is double antibody sandwich means here the antigen is bound from lower side by an antibody and from the upper side, and that means from both sides this antigen is sandwiched by two layers of antibodies. One layer is this and another layer is this. That is, this whole structure forms a sandwich. Okay, where the antigen is sandwiched by the two layers of antibodies. That is why it is known as double antibody sandwich ELISA or DAS. Okay, so this is the process of double antibody sandwich ELISA, and used for detection of antigens, hormone, drugs or steroids, etc. also in the plant virus. Okay, thank you very much.